gel. Or you're going to make the gel, you're going to run it on the gel. Okay, so this down here is the original wells, and this is the DNA ladder. It kind of got a little bit washed. This gel was probably not mixed thoroughly. Okay, if you watch the video, you'll see that I tell you to stir it and boil it, and then stir it, bring it to a boil. You got to do that 4 times and keep stirring it, because if you don't do that, this is what happens. If you have chunks of undissolved agarose in there, and it doesn't run neatly. Okay, but anyway, it's good enough that we can see this is the insert and this is the plasmid. And this is a good gel. There's a lot of material here. If this experiment had been completed, I am sure the ligation would have worked, because this group had plenty of material to do the ligation with. Okay? So again, we're going to cut out this insert, we're going to cut out the plasmid, and then we're going to do the gel extraction again. Right? You know how to do that. Place in the pre-weigh tubes, carry out the gel extraction kit procedure, same as before. Keep the plasmid and insert separate. Right? You need to know which is which. So label your tubes so that if you take your little column out, you don't accidentally put the column into the wrong tube. Right? Or you get them switched. Don't get them switched, because we need to know at the end, because we're going to add 3 microliters of insert and only 1 microliter of plasmid to the ligation. So we want to know which is which. Right? Otherwise, you want to have a lot more insert than plasma. Okay. Then, this time, since we're going to do the ligation, elute with only 15 microliters of EB buffer. Right? Again, wait 4 to 5 minutes after placing the buffer onto the white membrane before spinning it down. Right? Instead of using 20 microliters, we're going to go even further. We're going to use 15 microliters, because we really want as much material for that ligation as possible. Okay? And for the ligation, this is the list of components. We're going to keep the enzyme, the ligase, should stay in that blue freezer box, just like with the restriction enzymes. Okay? And you're going to put in this combination. This is 2x ligation buffer, so you can put 5 microliters, 1 microliter of T4 DNA ligase, and then 3 microliters of your purified insert, 1 microliter of your purified plasmid. Make sure that they're all together. This is a very tiny volume of stuff, so you don't want 1 drop of them up here and the rest of the drops down here, because if they don't mix, they don't react, right? Okay, so to get them all down there, spin it down in the centrifuge and make sure all the drips get all the way to the bottom. And then you can carefully tweak it onto the vortexer, and so that you spin that, mix that well, okay? And make sure everything's in the bottom, and then you can wait, right? So you only need to centrifuge for about 5 seconds to get all those drips of water down to the bottom, okay? Vortex, and if you see that it's not all the way at the bottom, you can spin it again for another 5 seconds, okay? And then wait 5 minutes, okay? And the last step is going to be the transformation, okay? So I did put a procedure on the web, the one that's called Competent Cell Protocol. For whatever reason, it doesn't open. I tried different ways of loading it up. It still doesn't open. So, but there is a molecular biology protocols link. Okay, and that's all the protocols in one collection, and the Competent Cells Protocol is in there. So you can get that out and make sure you've read it. The key with this is timing. Right? If it says 5 minutes, it means 5 minutes. If it says 30 seconds, it means 30 seconds. So you get out a stopwatch and measure each of those times exactly. Don't let the thing warm up. Each step is critical, and I will work with you, because usually by this time everybody's gone anyway, so it'll just be us, the molecular biology and me. So we will be finishing this up together. Okay? All right. So then, after you finish the transformation, you're going to pour that little tube onto a plate that I have already ready for you that has kanamycin in it. Okay? And then we're going to put that plate in the incubator and pray that the colonies grow overnight. Right? And that's the end of week 1. Right? So, assuming that you have successful colonies, and I believe that you can do it. Right? It's possible. Okay? I'm going to pick some colonies from your plate the following week and prepare overnight cultures from your plates. Right? We will keep one plate. we'll keep one plate with a streak of the colonies, right? And then we run those colonies. We're going to do a mini prep from that culture, right? There's a separate. I think I'm going to show it in the second one. But I have the mini prep video that you can watch, so you make sure that you get all of those steps done correctly. Okay, and then we need to check. After the mini prep, we are going to check for the insert in the plasmid by restriction digest. So we can take like 10 microliters of each mini prep and load them on a gel and see if we get an insert. And if we have the insert, then we know that we have done our ligation completely. It worked. Okay? If you get no insert, that means somehow you got an empty PET plasmid in there that didn't get cut, and it ended up in your ligation mixture, and it transformed the bacteria, and the bacteria could grow because it's got the kanamycin resistance on it, so you have no GFP. And that's typical with these ligations, that you get a stray, right? Anyway, so hopefully you did the ligation, everything worked. If you got the insert, then you're going to take that from that. Don't throw these away when you do the mini prep. Keep them on ice, okay? And then what we're going to do is we'll do a transformation, okay? We purify the plasmid from when we did the mini prep. So we ended up with a pure plasmid at the end of our mini prep. This is what we will use to transform the competent BL21DE3 cells. Only these will glow green, okay? The Novablue cells are lacking the T7 RNA polymerase, so they cannot transfer